coming to the second Storer lecture. I'm looking at the faces, and I think most everyone here probably already heard my first introduction on Guangshuo, but by formality, I definitely want to remind everybody, and even more deeply, I think everyone actually knows Guangshuo here from past interactions. But anyways, it's my pleasure to welcome Dr. Guangshuo Ou from Tsinghua University in China. This is a homecoming for him. Guangshuo was a graduate student here under Jon Scholey, who's now retired and unfortunately couldn't be here today. But Guangshuo knows very well many of the faculty in this room from his time as a graduate student. He was in the former CDB, now BMCDB graduate group. I would wager a bet that he might be one of the most distinguished graduates from the group, and I think we should hold him up as an extremely good example for the current students on what you can accomplish coming out of this graduate group. So Guangshuo, as I've just mentioned, did his PhD here. He went on to do a postdoc at UCSF under Dr. Vale, which is where we met. He graciously agreed to train me in the Vale lab for a bit, tried to turn me into a worm scientist. Everyone here knows that failed, but he was a great mentor. As more of my problem, I had no patience for injecting worms. I wanted to go right back to making proteins in the test tube. But anyways, I thank you for that. It was a great experience training under you. And then when he left Ron's lab, he went to China to start his own group, and he did a short stint at a university. I'm looking for the name of it here, but Institute of Biophysics. Institute of Biophysics, thank you. But then quickly was recruited to Tsinghua, which, in case you don't know, is one of the top biomedical research universities in China. And there he's really established an amazingly productive group at Tsinghua. He's now also listed as a vice president, so he moved his way up to the top of this esteemed university. So really amazing work. And his lab is a large lab that does a lot of breadth of science, but he's always maintained his love for the cytoskeleton, I would say. And that's what we'll hear about today. He's talked about forming ciliary microtubule structures. He's spent most of his career studying cilia. And so I'm really excited to hear this second lecture for the Storer. So thank you for coming this long way. Coming back, we've got nice photos of him sitting at his old graduate student desk, going around looking at all the old posters that might still be the same from 20 years ago. So it's quite fun to have him here. So with that, I'll let you take it away. Thank you, Guangshuo.
Thank you, Ray. I think I'm quite familiar with this room. This is the room I give my exit seminar, and I think this is the room that John gave his exit seminar, and I watched that video. And I think this is also the room I gave the Friday cytoskeleton meeting. I think that's amazing. You know, after the meeting, so Leslie, Frank, and Dan and Ken also give a lot of excellent advice in terms of project, in terms of career development. So today, I will going to continue with my PhD thesis work, but in a different way. So, you can use the arrow key. Arrow key. Where is it? This one. This one. Excellent. This work. Or not. We tested it.
Click the mouse first.
Sorry?
Click the mouse first.
Oh, okay, just click the Dan knows. He does give good advice.
We've never seen this slide.
Yeah, so here just quickly summarize. Give you a brief introduction. So cilia formation requires a process called intraflagellar transport. And the anterior grade transport motor equal kinesin 2 that was isolated from the Scholey lab by Ko in 1993. And this idea, so microtubule polarized, it's so basically the minus end is at the base and the plus end is on the tip. So anterior grade transport is powered by kinesin 2, and then at the tip, the dynein 2 is going to recycle the whole system. So today I'm going to talk about, how first we're going to talk about our ideas. How kinesin 2 going to load cargo and unload cargo. And the second is that how tubulin is assembled to make the doublet microtubule. And last I'm going to talk about our recent study or efforts trying to use AI-based pipeline, trying to see whether we can identify some previously unknown protein that are going to interact or contribute to this process. So that's I would say that's an ugly imaging. Well, because I still remember that's Dan's comments. So I showed this imaging to attend. I once used this slide to attend the International World Meeting. That's in 2005. And Dan said, "Oh, you have so many disastrous combination of color." Then later, I just changed to simple color.
Do you agree now, or you don't?
I agree. I agree. Sorry, that's all by myself. That's all my fault. So anyway, so I think the point that so as mentioned yesterday, so there are two kinesin called kinesin 2 and osmo 3 transport the cargo in the middle segment, and only osmo 3 transport the cargo to build the distal segment. Now, after we figure that out, we decide to use a genetic screen to identify the molecule that contributes to osmo 3 based distal formation. So I think that's in the summer of 2004. I screened. So basically, that screen kind of introduce an IFT marker to the existing known cellular mutant. So basically, it's not EMS-based screen. It's a genetic cross. It's so lucky. The first one I got, the first successful we got, shows a phenotype, phenocopy of osmo3. Look, the name is called DIFE1, which is a diphylline 1. So there are totally, at that time, there are 13 DIFE genes. So basically, I got them from CGC and I did the genetic cross, introduce an IFT marker. So at that time, I show get rid of DIFE1. The osmo3 cannot move, not even in the middle segment. So based on the genetic data, I propose DIFE1 can be an adapter or activator for osmo3 kinase. Based on genetics, that totally makes sense. Then also spend some time trying to purify the DIFE1. I think I indeed prepared the DIFE1 from bacteria, and then also prepared osmo3, but there's no activation. So it's biochemically, it's not activated, but genetically that shows the phenotype. It shows the phenotype, so they are in the same pathway and fits the genetic criteria. So after this paper published, actually I received a lot of criticism. What's your evidence? It's just genetic evidence. That's a big hurt for me because as a cell biology, it's not easy to do genetics. Then from different species, there's some different ideas for the function of the DIFE1. It's homolog. It's called IFT70. So I thought nobody going to believe my data. But after 12 years, I think I still remember that's 2017. I was invited by Tsinghua University, and she works in the Technical University, Technical University of Munich. And she wrote an email. We never met before. She wrote an email, asked me to visit his Department of Physics. That's something quite strange. I'm really bad in physics, but I was invited by Department of Physics. And I didn't reply the email in the beginning. But after a few months, Jon said I should reply the email and go to have a look what's going on. So the first time I met her, and the first sentence she spoke to me with me that I believe it was Nature paper. I was shocked. I'm self-doubt. What about the results? Then she told me something as illustrated here. So basically, at that time, this paper is almost accepted by Nature as an article. If you look at the Nature article, you look at these journals, this is the article that contains the few list data, the little amount of data probably at the time. Say this is just one figure. So we know, for each paper, each figure you have multiple panel, but this panel, so this figure is one single panel. It just turns out to be so important. So first, she told me, she confirmed DIFE1 by itself cannot activate osmosis kinase. I told her, I know that. And she told me she's trying to assemble a complex to activate osmosis. I just asked her, how are you going to assemble a complex? Because at that time, we know it must be an IFD particle. But if you think about the IFD particle, it has 22 subunits. How are you going to make a combination of these 22 subunits? It's just something like the like for the iPS cells, so that's called the Yamanaka factor. Put 4 different transcription factors to convert fibroblasts into iPS cell. But she just told me she succeeded and put 4 proteins together. Then she called it the quadripartite core complex called QCC. So right now, there's a AlphaFold, the structure information. It's easy to understand why you need to put that. But she started this project in the year of 2015. There's no clue, actually very little structural information about the IFD particle. So basically, she made the virus of each subunit, the bacterial virus of each subunit, and then mix them, do this kind of combination. And then eventually, as you see, this beautifully shown in this QCC can interact with osmosis 3 at 132 nanomole affinity, the constant number. But if you, without that one, there's no interaction. And she also did a beautiful single molecule study showing this indeed the activator. So this probably is the first kinesin, the activator for kinesin. So that's why it is so important. So then we recently just, with the aid of AlphaFold. So I'm talking about the structure right now today. It's all about based on the AlphaFold prediction. And it's not like the solid experimental data from Jordan. So I would say, many of them just hypothetical model. But this hypothetical model led us to do some functional study, make mutations in cell elegans or in human cells to test these possibilities. So right now, this is what at least we propose, we believe that dive 1, how to interact with the osmosis 3 tail, and we mute mutations, and we show, at least we get supporting evidence to show this model makes sense. Then for kinesin 2, well, this again is totally AlphaFold-based hypothetical model. So we found, so basically we did this kind of in silico screen. We found it's osmo 5, probably directly intact with CAP1. And we map out the interactions. So basically, well, this hypothetical interactions, and we made mutations. And the results, so basically, we start from C. elegans. So basically, using C. elegans, we create 2 knockouts, knockout osmo 5, knockout CAP1, and then transfect and make a transgene, introduce osmo 5 or CAP1 with mutations. And then we show, based on the rescue experiment, we show which residues are important. And it turns out it's a central interactions in just illustrate up this red chemical likely from these chemical bonds are essential to mediate the interaction. And we also show that this interaction might be conserved across species. And we test that using a human RPE1 cell. So in this scenario, we just start from the wild type, but we transfect the CAP1 with mutations. We show, if we mutate this residue, it cannot enter cilia, which means the docking or the entrance of kinesin 2 in the cilia require these interactions. Then after knowing how the kinesin 2 and osmosis 3 dock to the IFD particle, then this is of course interesting phenomenon. Also, I think Scholey Lab made an important contribution to its discovery. That's by Vale from Pittman Lab. I think the Brian visit Davis as an exchange graduate student, and the model is beautiful. It's called gradual turnover. The idea that kinesin 2 first transport the cargo, including osmosis 3, into the cilia, and then it gradually detached or detached or slow down or detached from the active particle, then the whole process, the active particle was handed over to osmosis mechanism. So we ask, what's the molecular regulation? How this was mediated? So for example, how to slow down or how to kick out the kinesin 2 from the active particle. And it's a tough question to address. This comes from our genetic suppressor screen. So we are doing a lot of genetic suppressor screen. I mentioned we work on the osmosis kinesin for 20 years, and we have collection of about 100 missense mutation. And for the summer school, that's a beautiful project. Each group was 2 students going to screen 1 missense mutation. In the beginning, we don't know what we're going to discover, but let's see. So this is one of the suppressor we cloned from one of the suppressor screen. So it turns out to be a Joubert syndrome protein, 26 protein. So this homolog mutate causes Joubert syndrome in human. And this is an interesting phenotype because you see lose the distal segment and the kinesin in this background. But if you mutate the JBTS26, the distal segment recover. Then we ask, what's the mechanism? Why? It turns out kinesin 2 labeled with cap1 now entered the distal segment. In terms of genetics, that's a perfect candidate we need to get. You see, in the wild type, kinesin 2 was limited to the middle segment, and the remove of one protein, now it enters into the distal segment. So then we further ask, what's the mechanism? So first of all, based on the localization, this JBTS26 protein localized only in the middle segment. Then we did the biochemistry, we show that this protein does not bind to singlet microtubule. It only binds to doublet microtubule we isolated from a chlamydia flagella. So as you illustrate here. Then we ask, how this protein interact, whether this protein has any interaction with active particle. And it turns out this protein interact with osmo5. Again, this is also a hypothetical model, but we have biochemistry as a backup. So then we did the biochemistry. So basically we transfect, make the bacterial protein or transfect the plasmid to the HAC293 cells and did a pull down experiment. As shown here, indeed, osmo5 intact with cap1 biochemically. And then in the same binding condition, if we add the JBTS26, you're going to weaken the interaction. Then we did some manipulation. Say in the wild type background, let's overexpress JBTS26. We show it's going to slow down the kinesin 2 because our model is that in the middle segment, kinesin 2 and osmosis rate slow and fast move together. So it moves at an intermediate speed. But if you overexpress JBTS26, kinesin 2 detach from the active particle, you're going to move at its own slow speed. And if that's true, then look for the osmosis rate. Osmosis rate now is going to increase the speed. So in the wild type, it's 0.7. Now it moves at 0.8, 0.9 micron per second. So that fits the model. So based on these results, we propose this JBTS26 protein that localized specifically in the middle segment because it only interact with the doublet microtubule. So it cannot go to the distal segment. And because it competes with kinesin 2 through binding to osmo5. So in this competition is going to let kinesin 2 detached from the active part. Then to provide some insights into this handover. So the kinesin 2 now gradually detached from the active particle.
Does it bind microtubules directly? Is
What?
that-
GBTS.
Yes.
Yeah,
Oh, so
Yeah.
it's a microtubule binding.
it's
protein.
it's doubly a microtubule binding protein. It does not bind to single layer microtubule. Kind of surprising. We didn't expect that result. So then what happened for OSM3? Right? So OSM3 entered the cilia, but it's not fully activated. So there are two possibilities to inactivate OSM3. One is folded conformation. And the second is probably some modification to the protein. So we decided to use the fluorescence lifetime microscopy to measure the conformation of OSM3 in the cilia. So that's a strategy. So basically, if there's a flag, the lifetime, they're going to change the lifetime. So as you can see here, the color looks quite different. But this is the control. So basically, that will be constant flag. And then if we tag them both ends, one end with reference protein, the other C terminal with GLP. If there's a flag auto-inhibited, that would be reduced the lifetime. So from this measurement, we surprisingly found, using this cartoon to illustrate, when OSM3 enters cilia, it's already in the open conformation based on the change of the fluorescence lifetime. Then the question is that there must be some regulatory mechanism. You need to inhibit the activity of kinesin 2. Sorry, OSM3 kinesin. We have been working on cilia field for quite a long time. We know some kinase is involved. For example, in this scenario, there's two types of pneuma family kinase one called NEKL3. Because in the genetic screen, we constantly get missense mutations. We never get a stop codon because Dave Fei worked on that for decades. If you delete that, then the larva is lethal. And so, so we suspect we always get the weak loss of allele for NEK3 that evolve in this process. We can get a non-allele for NEKL4 that evolved in the cilia process. But what's the mechanism is actually unclear. So then we use a non-allele of NK4, and then we also use the somatic genome editing technique to make a conditional allele of NK3. Now you can see so that stands for OSM3. In the double mutant, OSM3 has enriched signal or has an even signal in the middle segment. Then we further use a PCRG1, which is a protein bound to double mutant microtubule, to ectopically bring the NEK3, this kinase, into the middle segment. Because normally it's localized in the transition zone or the dendrite. And then if we put them into the middle segment, you're going to generate cellular phenotype, reduce the cilia. And if we, as a control, that D150A, that's a kinase static version that doesn't affect the localization activity of OSM3. Then further, we use biochemistry. We identify there's a S96 phosphorylation site within the motor domain of OSM3. So then the idea that so the R10, these are the residue I talked about yesterday. That's a mutation in the KIF1A, and that's important for the hydrogen bond with S96 to stabilize this ATP binding pocket. So then we use an in vitro single molecule assay, say for the OSM3 G4E, the full-length protein move along the microtubule. But in the same reaction, if we add a kinase, just 2 minutes, completely stop the motility of OSM3. Then we did a phosphomimetic or phosphodad experiment, and we show indeed it is phosphomimic. You're going to disrupt the cilia formation. And based on these results, so we further identified which phosphatase that are going to remove the phosphorylation, the phosphor group. So it turns out to be PP2A protein in C. elegans called LAT92. Again, N92 from the name is embryolally so. Unfortunately, we isolate weak loss of function allele. It's viable, but cilia is defective. And we also, I think we also make the condition shown here that we can rescue, and then we it can counterbalance with a knockout, conditional knockout cilia phenotype by NEK3. And again, if we use ectopic expression system to put the phospholase into transition zone, which means we dephosphorate the phosphorylation in that site. As you can see, the osmos 3 start to move earlier. So based on this information, we propose when osmos 3 entered the cilia, it's in the extended conformation, but it was phosphorylated by NEK in the transition zone or in the dendrite. So the motor is not fully activated. And then because of the function of that phosphatase, LAT92, remove the phosphor group, so it can activate osmos 3. So it gradually start to move by itself at full speed. So after we learned how the motor protein coordinate and how they coordinate at the molecular level, then it's a cargo. So so based on the previous study, we know the IFT particle that dock to IFT, the tubulin is one of the most important cargo that IFT transported to build the doublet or singlet tubulin. So for the tubulin, we know is transported by the interaction of the tubulin with IFT 72 and 81. And we had several years ago, we have a paper that show this tubulin interaction with IFT 72 with IFT particle that depends on di5 phosphorylation. Di5 phosphorylate IFT particle that release the tubulin at the tip. Now, when the tubulin arrives at the tip, then one of the big challenge is that cilias form this two-segment cilia. One is a doublet microtubule, and only a subfiber extends. Then the question is that how you can precisely maintain doublet and the singlet. that comes from a surprising discovery when we look about the microtubule inner junction protein. So yeah, so basically A in the doublet, A has 13 protofilament, but B only have 10. Originally we thought, oh, that's 11 protofilament. But the 11th position is not tubulin. It actually turns out to be two in the inner junction protein called PACRG and FAP20. And it's quite intriguing. PACRG does not express in the C. elegans sensory neuron. It's expressed in several other neurons. So the genome has PACRG in C. elegans genome. C. elegans genome has FAP20 for that protein. So then our lab actually spent a lot of efforts trying to do the high priority freeze of C. elegans and do the cryo fit milling to generate thin lamina to visualize the subcellular structure in situ. And this is what we found. So we eventually found the doublet microtubule from using cryo-electron tomography. And then we analyzed this region. You see, this region is actually empty. So we saw this is a model that we proposed because of lack of the PACRG. So the connection of the doublet microtubule, the ceiling of the B tubule only relies on FAP20. And if this model is correct, if we knock out FAP20, we should expect the open B tubule. So then we did an EM that show indeed in the FAP20 knockout, the B tubule is not sealed. However, if we knock out the PACRG, it's totally fine. You still get beautiful doublet microtubule. And we thought, whether we can overexpress ectopically express PACRG in second-facilitated sensory neuron. Let's see whether we can rebuild we can build the doublet microtubule in the distal. So this is what we did. Indeed, so basically we transfect them into so make a transgene overexpress ectopically express PCRG1 in C. elegans. And I think in this region, in the distal region, so in the wild type, we should not see any doublet microtubule. But now we can clearly build the doublet microtubule. Then after we know how they differentiate, the next question is about how to form the doublet microtubule in general. So this has been done, since 1980. At that time, the protocol is quite straightforward. Use an enzyme called substinin to digest tubulin. You're going to remove the tail of the tubulin, then it can form tubulin. It can form doublet microtubule in vitro. Then in C. elegans, we use genome editing to systematically delete the tail of alpha tubulin or beta tubulin. We have not completed all of them, but quite a few of them. And based on this mutant, we use a we show the technical message is that if we only delete alpha tubulin tail, we will be able to detect the doublet microtubule in the distal, but not the deletion of the tail of beta tubulin. So as shown illustrated right here. Then we perform the molecular dynamic simulation. Under these results that show that delete alpha tubulin, it allows the attachment of the B, the tubulin from the side of A tubule, but delete the beta tubulin, alpha tubulin tail still keep the tubulin away from the side wall of A tubule. Then we decide to do the biochemistry to show whether indeed A and B has some distinct function in the formation. We generate recombinant alpha tubulin, beta tubulin, with or without tubulin tail. Then we use a fluorescence assay that shows, so first we generate Goldman microtubule, then we add this recombinant GIP tail, the tubulin tailless tubulin into the system. So as you can see, delete the beta tail, it forms a microtubule, forms a new microtubule in the end. But if we delete alpha tubulin tail, we find some microtubule formation or green fluorescence along the existing microtubule. And if we delete double, then we get even higher efficiency in the green fluorescence formation or enrichment in this region. Then we use a TEM, a negative staining EM, to show indeed or confirm what we found for fluorescence imaging. The delete of the alpha tubulin tail is going to allow the formation of the doublet microtubule in vitro. And then the question is that, what's the physiological components that help for the recruitment or the help the formation of the doublet microtubule? So as you probably heard, this is a recent study from the cryo-electrotomography analysis of doublet microtubule structure. So there are a bunch of FAP protein identified. And we found a very interesting one called FAP53. So this protein sticks out from A11. And from the cryo structure, it seems it intact with B0 protophylline. We thought, oh, this could be one of the candidates. Let's think about it. Whether FAP53 can stick out from the existing B tubule and then dock or help the formation of the B tubule around A. So again, using molecular dynamic simulation, we show indeed if we add the green FAP53, there are two possibilities. One is that FAP53, if it's on the bottom for the B, for the formation, for the adding of the B, then it has two functions. it pick out or move away the inhibitory role of alpha tail. And it seems that I also can act as a docking site for receiving the B tubule from this side. But for the top, it's less efficient. So then we decide again to use the in vitro reconstitution to see whether we can indeed form the doublet microtubule in vitro. So we made recombinant FAP53 and using the standard assay, so basically form the microtubule and add FAP53 and then add the tubing again. So that's what we found using cryo-electrotomography. We show this is intact microtubule, and we found the formation of the B fiber of the B tubule around the A. Then because for cryo-electrotomography, the throughput is quite lower. You have to spend one day and get a few imaging. But here, we also use the TEM to use a unit sectioning. We can easily quantify more than 1,000 of the microtubule. We found indeed it forms some structure that shows the formation of the doublet microtubule. And because we didn't add FAP20, so it totally makes sense. It's not fully curved. It's not fully full. Yes, so then we ask whether we can generate doublet microtubule in HeLa cells. In the cytosol of HeLa cells, we know HeLa cell does not form cilia. Even if you do the serial starvation, it will form the cilia. Then we transfer both FAP53, and it does not show any microtubule localization. But if we co-transfer FAP53 and FAP20, now in some area, we found there's co-localization of these two proteins with a microtubule. Again, we perform the TEM. We show indeed for the control, it's always single-layer microtubule. But if we transfer two of them, at least we can detect some of them. Looks like the doublet microtubule. Then we ask, what happens if we do the same thing in C. elegans dendrite? We know C. elegans dendrite is all about single-layer microtubule. Then we did the similar transfection or transgenic animal, and in the dendrite, because we do the serial section from the top of the nose, we know where the dendrite can be. So you can see we found clearly doublet microtubule ectopically formed in the dendrite. So all this data that they just proposed, just allow us to propose, probably it's an FAP53, help the physiological formation or reconstruction of the doublet microtubule in vivo. So finally, I'll talk about something, whether we can find a novel regulator for microtubule. So we know the first microtubule binding protein is tau. That's for about 50 years ago. During the past 5 decades, whether we can still find a new microtubule protein or completely unknown microtubule protein, I don't know. But my student start with a turbo ID. It's a fancy technique. Everyone uses turbo ID. The idea that he want to purify the protein on the surface, on the outer surface of the microtubule, or even inner surface of the microtubule, so-called MIP, microtubule inner protein. Then everyone knows the problem of the technique. It's easy to perform, but you always got thousands, 1,000 or several hundred hits. 99% of them are garbage. Everyone agrees that. So then we got a whole list. Then I asked the student, please do not do any experiment. Let's do an AlphaFold prediction. Let's check, just put the tubulin sequence and your protein sequence to AlphaFold. Just spend up 20 minutes. Let's have a look whether it looks like the microtubule binding protein or not. And then we continue. So based on this strategy, I think he identified one that there's no publication from this kind of mass spec. Then another student who is really good at AI-based modeling, and he asked, whether we can do something even crazy. So in this human genome, we know that it's about 100, that kind of range, that 100 known microtubule binding, at least in that range. Then they got a structure information just like that, do one by one, about 100. And then undergo a process called distill. Now I understand what's a distill. Basically the idea that from this point of view, please forget about the 3D dimension, forget about everything about the chemical bond interaction. In the beginning, I don't believe that. So then they just map out what's the binding site based on the 3D conformation. Then use that information to build a large language model. Using this large language model, they can quickly predict, look at the time, 20 minutes to 0.02 seconds to predict whether this is a microtubule binding protein. Well, because we have data sets, we have experimental data sets, so the model works so well for the training data sets. Then we spend half an hour to screen 20,000 human protein. It's really fast. And we got a bunch. We got a lot top list. Of course, we got all the known ones. And we just want to focus on the one only have ORF. Basically, look at the database. There's no study, there's no information, just ORF. We ordered 3, and for one of them, just beautiful microtubule localization pattern. So I started to believe large language model might be something really useful because it identified one from there. And then we also ask why, previous people didn't find this microtubule binding protein. So probably Frank knows all the history. So basically for the microtubule binding protein, they use a brain tissue or xenophil extract, but this gene does not express in the brain, only express in the testes. So it could be very likely a sperm-specific binding microtubule binding. After that, we want to move further step. Well, so this is really the progress. It's a loose end, I would say. So I don't understand much about the slides. My student sent me a slide. I just want to show you what we want to do. So we want to design completely novel microtubule binding protein that does not exist in the world. Then the only thing we need to do is to constrain how long you want. Because usually the microtubule binding protein, the microtubule binding domain is about 100, the 100 amino acid. Then for 100 amino acid, how many possibilities? There are 20 natural amino acid. That'll be 20 to 100. So that's a number. So my student told me, let's just random generate amino acid sequence. There's no input, just random generate them. And then the 2 criteria, they score 2 things. One is they use Dave Baker's peptide to score whether this peptide reads like a protein. So we know if you generate 100 glycine, that cannot be a protein. So this part I can understand. And the second part is that since we have a predictor, we can score whether this protein is a microtubule binding protein within 0.02 seconds. Okay? So we can combine that information to this generator that is score. So basically we got 2 score. And afterwards, I don't know what's going on. So he told me there's some annealing or anemia or something. So basically, so they're going to repeat the step 170,000 steps and then get what they need. So apparently, it depends on how much computation you do, right? So if you do, then you get one. So basically, we start after we create a 20 something. 20 peptide shows the interaction with the microtubule based on the generator. Apparently, in this scenario, we cannot use AlphaFold. So AlphaFold will not show any binding with microtubule because this protein never exists in the nature. So there's no training. So they are not in the training data sets. So then we decided to synthesize all of them. We synthesized 20 of them and tag them with GIP. This is what we found. First of all, looks for these 6. It has filaments, right? It's not random. It's not GIP diffusing. It shows filaments. But who knows? Frank, do you know what kind of this filament?
It says active filament.
Unfortunately, they're all stress fibers. So basically, we also have the staining. So basically, fluoride is staining the actin filaments. So that's something we still don't understand. So we have a predictor for microtubule, but it got so many beautiful actin binding.
There's an explanation for this.
I have no idea.
The charge.
The
The
charge.
surface charge of the microtubule was negatively charged, and it mimics actually, even tau finds actin.
Binds to actin.
Very well. Very well. So it makes actually a lot of sense. And it depends on the cell that you have,
Yeah.
how much actin it has versus how much tubulin.
Mhm.
These cells have a lot of actin, so it takes away all the pool of protein.
That's great. Yeah.
So you have to find a cell that doesn't have actin, and it might find microtubules there.
Let's see. Yeah, so probably, yeah, thank you very much. So that's always
Sure.
useful to speaking in Davis. You're
So if
gonna
you
get-
Franklin treat these, will it then bind microtubule?
we have not done that. Yeah, but I told my students they should do this. But we have the phenolidine staining, is beautiful colocalization. Yeah, so I guess I would like to end my talk here and thank this funding agency and my collaborators. I'm happy to take any question.
I have a question.
Yeah.
So on the Joubert protein, the regulated kinesin 2, so does that imply that it must be spatially regulated with respect to the protofilament?
Yes.
So it implies that the kinesin is moving on a single protofilament? Is that right?
Well, for the kinesin 2, definitely not like that, because the kinesin 2 can take the side. We show that in vivo and also in vitro. My collaborator Tsinghua University is a purified kinesin 2 and also use a microtubule that you hang in the solution that show kinesin 2 can turn around.
But that's on a
step.
but that's on a naked microtubule.
Yes,
So
but in
in a-
vivo, we also use a super resolution imaging that show the vivo. Actually, kinesin 2 also take the side steps.
Okay.
So it's definitely not limited to any single protofilaments, probably can go anywhere.
So is the Joubert protein then also on multiple protofilaments, or how do you?
That's something we don't know yet. So originally we thought what's the specific interaction will be the inner junction protein, say whether it interact with FAP20. If so, that would be beautiful explanation to explain the specifics. But actually, it doesn't interact with FAP20. We did about chemistry show.
And what you showed, it's a folded protein, no?
FAP20.
No, that I know. I'm talking about the Joubert syndrome protein.
Yeah.
It's folded, right?
Yeah, it is folded.
So
Yes.
then the explanation may be that it actually aids the binding of the kinesin to the microtubule. It might be an enhancer of binding during the walking step.
Mm.
I mean, it'd be interesting to see what exactly the
If you
explanation is
bind to
structural.
kinesin 2.
Yeah.
Yeah, that could be.
In the FAB20 one where the B tubule comes off,
Mhm.
why aren't more protofilaments growing on that and why doesn't it complete a whole loop?
Yeah, that's a good point. I have no idea, but we also have the imaging. So actually, when the entire microtubule, there's one loop, there's another loop. So probably it's indeed a continuation of the formation of the protofilaments.
So along those lines, so with FAP20 and FAP53,
Mhm.
you express those in HeLa cells and you could get a full doublet.
Yeah.
You didn't show that you can just use those purified proteins in vitro to get a full doublet.
We have not done that.
Okay,
Yeah.
so I was wondering if it's sufficient in vitro or if there's
Mhm.
something else that you need in HeLa cells.
Yeah, probably additional protein in killer cells to get the full doublets. Yes.
Question about that, the last part. So technically, ideally, you can also design the de novo proteins to bind microtubules, actin, filament, whatever you want. You can also design those to cross-link them together.
Yeah.
So,
In theory, we should be able to find something.
Questions? I have maybe one more about the kinase activity regulating the kinesins. So again, sort of spatially thinking about it, my understanding of the model is that you have the two kinesins and they have different speeds. And so on that first segment, you get sort of an average speed. Is that right?
Yeah.
But that would imply that they're always active, right? But your new model suggests that the faster kinesin is actually inactive by the kinase.
By the kinase, that's right.
So does that fit with the velocity data that?
Yeah, it's just because of this kind of regulation, probably it's called gradual regulation. You look at the localization for this kinase and the phosphatase. It's not like, say, completely restricting some area. It shows the enrichment in transition a little bit in the middle segment. So probably it depends on how much kinase they are active in that region. I think, yeah, probably in vivo it's a mixed population of inactive and active. I think. Yeah.
I have the same problem with the gradual changeover, but yet the speed, maybe you presented that the whole segment has the same speed. It's not like it's,
That's oversimplified. So this Peterman lab gives more accurate measurements. So it's a gradual change of the speed.
And do you have any ideas about the spatial regulation of how the axoneme switches from doublet to singlet? So what causes that trend? What causes that?
I think that's the JBPS 26 protein. That's one.
So how does it know to stop right there? Like,
yeah, so the idea is that probably, yeah, that's a good point. So the singlet microtubule, the doublet microtubule, the localization of that protein. And also probably it's a kind of gradual loss of kinesin 2. So because if we look at the fluorescence distribution of the kinesin 2, actually enriching the transition zone middle segment in the initial stage of the middle segment, and then gradually becomes dimmer. Yes.
So when you are trying to predict the map for the microtubule binding sites, so in the AlphaFold, so you are putting the sequence of alpha and beta tubulins, right? And you are trying to find all those hits which actually binds. So how many like alpha and beta tubulin like you are putting in inside?
Yes. So right now we put 2 dimers.
So that's not enough. Like, do you think it's
Not enough. We definitely miss, miss the interaction between these 2 protofilaments. You're right. But it depends on how, the calculation power, how, if you think about what's the size of the GPU to run the program.
great. Well, if there's no more questions, let's thank Guangshuo for.